Hello friends, welcome to SRS by tutorial. I am Samiranjan. In this video, we will discuss about beta oxidation of saturated fatty acid with even number of carbon atoms. What are fatty acids? Fatty acids are simple lipids. They have a carboxylic group and a hydrocarbon chain. There are two types of fatty acid, saturated fatty acids and unsaturated fatty acids. Saturated fatty acids do not contain double bond in the hydrocarbon chain, whereas unsaturated fatty acids contain double bond in the hydrocarbon chain. What is beta oxidation of saturated fatty acid? It is the oxidation of fatty acid on the beta carbon atom with the removal of two carbon unit as acetyl coenzyme A from the carboxylic end. The beta oxidation of saturated fatty acid involves three stages: activation of fatty acid in the cytosol, transportation of fatty acid into mitochondrial matrix, breakdown of fatty acid by oxidation. Now come to the activation of fatty acid in the cytosol. This reaction is catalyzed by acyl coenzyme A synthetase, which is present on outer mitochondrial membrane. This enzyme catalyzes the formation of a thioester link between carboxylic group of fatty acid and thiol group of coenzyme A to yield a fatty acyl coenzyme A. This reaction is coupled with breakdown of a molecule of ATP to AMP and pyrophosphate. This reaction occurs in two steps and involving a fatty acyl adenylate intermediate. This is the overall reaction of fatty acid activation. The release of pyrophosphate make this reaction irreversible. The second stage is transportation of fatty acid into mitochondrial matrix. The inner mitochondrial membrane is impermeable to fatty acid. A specialized carnitine carrier system transport activated fatty acid from cytosol to mitochondrial matrix. The fatty acyl coenzyme A react with hydroxyl group of carnitine to form fatty acyl carnitine. This transesterification is catalyzed by carnitine acyl transferase 1 present in the outer membrane of mitochondria. The fatty acyl carnitine then move across the outer membrane to intermembrane space. The fatty acyl carnitine enter into mitochondrial matrix by facilitated diffusion through the acyl carnitine transporter of the inner mitochondrial membrane. Then the fatty acyl group is transferred from carnitine to intramitochondrial coenzyme A by carnitine acyl transferase 2. This conversion regenerates fatty acyl coenzyme A and releases free carnitine into the matrix. The carnitine re-enter into intermembrane space via acyl carnitine transporter. Then the carnitine again involved in transportation of another active fatty acid into the mitochondrial matrix. The final step is breakdown of fatty acid by oxidation. This process includes four enzyme catalyzed reaction: oxidation of fatty acyl coenzyme A, hydration of trans-enol coenzyme A, oxidation of β-hydroxyacyl coenzyme A, and thiolysis. Now come to oxidation of fatty acyl coenzyme A. The fatty acyl coenzyme A undergo dehydrogenation to produce a double bond between alpha and beta carbon atom to produce trans-enol coenzyme A. This reaction is catalyzed by acyl coenzyme A dehydrogenase, which is a FAD dependent flavoprotein. The electron removed from fatty acyl coenzyme A are transferred to FAD and reduced into FADH2. Then hydration of trans-enol coenzyme A. In this step, a molecule of water is added to the double bond of trans-enol coenzyme A to form L stereoisomer of beta-hydroxyacyl coenzyme A. This reaction is catalyzed by enol coenzyme A hydratase. The next step is oxidation of beta-hydroxyacyl coenzyme A. The beta-hydroxyacyl coenzyme A is dehydrogenated to produce beta-ketoacyl coenzyme A. This reaction is catalyzed by beta-hydroxyacyl coenzyme A dehydrogenase. During this process, NAD plus act as a electron acceptor and reduce into NADH. The final step is thiolysis of beta-ketoacyl coenzyme A. The beta-ketoacyl coenzyme A reacts with a molecule of coenzyme A and produces acetyl coenzyme A with 2 carbon less fatty acyl coenzyme A than the previous. This reaction is catalyzed by beta-ketoacyl coenzyme A thiolase. The 2 carbon less fatty acyl coenzyme A re-enter the beta-oxidation cycle. This process will continue until the fatty acid completely oxidized into acetyl coenzyme A. Let's take an example of beta oxidation of palmitoyl coenzyme A. The palmitoyl coenzyme A undergoes 4 enzymatic reaction of beta oxidation to produce myristoyl coenzyme A and acetyl coenzyme A. In one cycle of beta oxidation, one molecule of acetyl coenzyme A, two pair of electrons, 4 protons are removed from the fatty acyl coenzyme A. Hence, the equation for one cycle is as follows. The myristoyl coenzyme A undergo another cycle of beta oxidation reaction to produce second molecule of acetyl coenzyme A and lauryl coenzyme A. The complete oxidation of palmitoyl coenzyme A required 7 cycles of beta oxidation. It is the overall reaction of complete oxidation of palmitoyl coenzyme A, which produces 8 molecule of acetyl coenzyme A and 7 molecule of FADH2 and NADH. Energetics of beta oxidation. Each molecule of FADH2 produced during oxidation of fatty acid donates a pair of electron to electron transferring flavoprotein of the respiratory chain. About 1.5 molecule of ATP are generated during the transfer of each pair of electron to oxygen. Hence, 1 FADH2 is equal to 1.5 ATP. Similarly, each molecule of NADH delivers a pair of electron to mitochondrial NADH dehydrogenase. Then subsequent transfer of each pair of electron to oxygen result in formation of 2.5 molecules of ATP. Hence, 1 NADH produces 2.5 molecule of ATP. Hence, 4 molecules of ATP are produced during removal of 2 carbon unit in one cycle of beta oxidation reaction. Then 7 cycle of beta oxidation produce 28 molecules of ATP when FADH2 and NADH produced in oxidation enter into the electron transport chain. This is the overall reaction of complete oxidation of one molecule of palmitoyl coenzyme A. The acetyl coenzyme A produced from the oxidation of fatty acid can be oxidized in citric acid cycle. During this process, one molecule of acetyl coenzyme A converted into 10 molecule of ATPs. Hence, 8 molecule of acetyl coenzyme A converted into 80 molecule of ATP. Then, the complete oxidation of palmitoyl coenzyme A produces 108 molecules of ATP. During activation of the palmitate to palmitoyl coenzyme A breaks both the phosphoanhydride bond of ATP. The energetic cost of activating a fatty acid is equivalent to 2 molecule of ATP. Then net gain per molecule of palmitate is 106 molecule of ATP. This is the summary of beta-oxidation of saturated fatty acid, in which the fatty acid is activated by acyl-CoA enzyme synthetase to produce fatty acyl coenzyme A. Then the fatty acyl coenzyme A is transported into mitochondrial matrix by carnitine shuttle system. Inside the matrix, the fatty acyl coenzyme A is oxidized through 4 enzymatic reaction to produce acetyl coenzyme A and a new 2 carbon short fatty acyl coenzyme A than the previous one. In the next video, we will discuss about